My name is Nandan Raman, and our project is Influence of Natural and Man-Made Substrata on Intertidal Community Structure. Abstract. As humans continue to expand and build new structures and pollute water bodies, it is vital that we ensure the persistence of coastal life forms and ecological communities. Focusing on 5 coastal sites, this study utilized the transect and quadrat sampling methods to compare species diversity and community structure on both natural rock substrata and man-made structures like jetties. Observing both sessile and motile organisms, the data is analyzed through the ANOVA method to see the difference in species richness across the different structures. This study aims to understand how human-made coastal modifications have influenced marine biodiversity. We learned that there were differences in community structure with an increase of species richness in natural substrata. Hypothesis. I hypothesize that community structure will differ between natural and human-made substrata. Methods and procedures. Sampling methods. Our study involved the transect method, where we created a 10 by 10 inch PVC quadrat. Then, to collect our data, we randomly place this quadrat throughout each site. On the left is one of our quadrat pictures we used, and all the data was collected at a 1.5 feet tide height to ensure consistency. Roving study. We also conducted a roving study for 5 minutes at each site, where we looked for as many living organisms, and this data was incorporated into our data. We used a 10 by 10 grid with 100 randomly scattered dots on it and overlapped it with each photo to count of all the species present at each dot. Then we transferred this data to a spreadsheet and employed the ANOVA method. It produced many statistics, but the one we directed our attention to was the p-value, which helped us determine the significance of the differences across all of our sites. Conclusion. 4% algae, the ANOVA method produced two p-values, 0.16 and 0.95, both greater than 0.05, meaning that there was not a significant difference, although the number of species in the two p-values were lower than 0.05 and show that there were more species present in natural sites. While the community structure was partially similar, this data allowed us to conclude that there were some differences, especially in the number of species and species richness. Below you can see my data table. Thank you. Again, and here are my references for my data.